Mittali from group 319, and here I am presenting my topic non-linear dynamics of membrane. In this topic we will discuss about the dynamics of membrane, like low mass balance, diffusion, facilitated diffusion, potassium mediated transport, active transported. So these are the topics, like in a low mass balance, inside or outside we will discuss. Diffusion is very slow. Facial diffusion is like carrier proteins. Protein mediated transport is very selective. Active transporter uses ATP like sodium potassium. ATP is pump. They uses energy, they wanted energy. This is called active transportation. And there is passive transport uses, they do not require energy. Membrane dynamics. Cell membrane structure and function. Membranes form fluid body compartments. Membrane as barriers and gatekeepers, like vesicle, transepithelial osmosis, distribution of water and solutes in cell and the body. Chemical and electrical imbalance are also resting membrane potential, membrane permeability and changes. Next is vesicular transport. In vesicular transport, movement of macromolecules across cell membrane, like phagocytosis, endocytosis, exocytosis. In phagocytosis, specialized cells only. In endocytosis, like pinocytosis, receptor mediated endocytosis, like cellular photocytosis. And next is exocytosis. In the phagocytosis, mainly the cells are kill the foreign materials. We will discuss about the phagocytosis. Phagocytosis requires energy. This is a cell engulf particle into vesicle via pseudopodia formation. Example: some WBCs engulf bacteria. Vesicles form are much larger than those found by endocytosis. Phagosomes fuse with lysosomes. This is a process which all are the required the energy. By the energy this process is completed. WCs kill the foreign material in engulf. Next we will discuss about the endocytosis. Endocytosis also requires energy. There is no pseudopodia membrane surface indeed. These are the smaller vesicles, and non-selective and selective both are this function. In the non-selective, phagocytosis for fluid and dissolved substance. In selective receptor mediated endocytosis via calithrin coated pits. Example: LDL cholesterol and family hypercholesterolemia. Photocytosis via cellular. There is a diagram. In this diagram we will discuss in this diagram many things are represents, like intracellular fluid, extracellular fluid, coated pid, ligand, receptor, membrane, recycling cell membrane to lysosome or Golgi apparatus are present. Next we will discuss about the exocytosis. In the exocytosis, intracellular vesicle fuses with membrane. This process also required energy, like ATPA and Ca2 plus. This is uses in large lipophobic molecule secretion receptor insertion. In this diagram there is a Golgi body and present and represent the transport vesicle, secretory vesicle, membrane, protein transport vesicle, lysosome and peroxisome also present transfer vesicle. In this process waste removal are showing. Next we will discover the movement through epithelial transportation. In this transport uses combination of active and passive transport. Molecule must cross two phospholipid bilayers. Apical and bilateral cell membrane have different proteins, like Na plus glucose transporter on apical membrane and Na plus K plus and ATPase only on bisolateral membrane. Next one is transcytosis. In the transcytosis, endocytosis is functional vesicle transport, and vesicle transport is functional exocytosis. In this, the move large proteins intact. Example: absorption of maternal antibodies from breast milk and movement of proteins across capillary endothelium. There is a diagram present the exocytosis cells and the capillary endothelium. In plasma protein are and red blood cells are also present. Intracellular fluid all are present, and they are showing the Next is distribution of solutes in body depends on selective permeability of cell membrane transport mechanism available. Like water is an osmotic equilibrium, free movement across membrane, ions and most solutes are in chemical disequilibrium. Example NAK, sperm. Electrical electrical disequilibrium between ECF and ICF. Next is a diagram in this way diagram showing the osmosis process. Or osmosis process is the movement of water down its concentration gradient, like oppose movement of water across membrane. Water moves freely in body until osmotic equilibrium is reached. Equilibrium process in which the both the sides are water and molecules are equal and there is a osmotic process is equal. So this is the presentation. Thank you.